Okay, the camera, when I was going over dehydration synthesis, the camera died or did something. I guess if I don't change the focus or touch something on it, it turns off after a certain amount of time. So I'll try to figure out, this is a new camera for me and everything like that. One last thing I did want to talk about in terms of carb, sugar, saccharides. We use glucose and fructose to make sucrose, okay, a disaccharide. One other disaccharide that you have to know besides sucrose, and that sucrose is a combination of glucose being bound to fructose by a process known as dehydration synthesis, okay? The last one is maltose. Maltose is another disaccharide made from two monosaccharides, but this part is glucose bound to glucose. All right, so you have basically oxygen here, and you have right here, and this is glucose, which is a 6-carbon ring, and glucose, 6-carbon ring, 1, 2, 3, 4, 5, oxygen, 1, 2, 3, 4, 5, oxygen, so 6 sides. And what did that look like? Well, it's right here. So here's my glucose right here. There's the single oxygen holding them together, and here's my other glucose molecule. So they'll lay this right here, 6 ring, 6 ring, maltose. Okay? All right. Time to move on to lipids. Otherwise known as fats. So fat, like carbohydrate, is the vernacular. Lipid is the scientific name. For sugars, it's saccharides. So you have saccharides and sugars. Here we have lipids and fats. Okay? So what makes up a fat? All right. First of all, you need to identify two things. Something called the glycerol head. Glycerol. Okay? And I added in the word head. Okay? Glycerol. Okay? It is a 3-carbon compound. Okay? For glucose, it was a 6-carbon ring. For fructose, it was a 5-carbon ring. This is a 3-carbon head. Okay? Well, if you have a head, what do you have attached to a head? Tails. Okay? In this case, the glycerol head, having 3 carbons, has 3 tails. These tails are called fatty acids. Okay, fatty acids. Can you see that? Yeah, it's kind of. What's the construction of a fatty acid? Well, it is a long chain of carbons and hydrogens. A long chain of carbons, and now I'm adding hydrogen. Okay? Let's have you get a really good close-up look of that. There we go. Keep that in focus. Okay. I don't want to have to draw 3 of those. Okay. So we have fatty acids. Okay. A long chain of carbons and hydrogen, which we call a hydrocarbon chain. Hydrocarbon chain. All right. Now, chemistry. Every carbon that exists needs to have 4 bonds, okay? 2 pairs of electrons. This carbon has 1, 2, 3, 4, or 2, 4, 6, 8, okay? Valence electrons. This carbon, 1, 2, 3, 4, that works. This carbon, 1, 2, 3, 4. There has to be something bound at the top, one at the bottom, to the left and to the right. Notice that our problem is at the end. We are missing what? That's right, we're missing a hydrogen. Let me identify that by signaling it out. 1 carbon, 3 hydrogens. 1 carbon, 3 hydrogens. Okay? Remember our functional group that we didn't talk about? The functional group was, brain's going, methyl. Okay? What is the methyl group? What does that do? Well, the methyl group acts as bookends. It acts as an end for a long hydrocarbon chain, among other things. But for what we're studying, this is our methyl group. Okay? This right here, CH2. In fact, we have a bunch of CH2s bound together all the way down here. CH2, 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 CH3. Okay? All right, if every single carbon has 4 bonds around it, as each one of these does, we call that a saturated fatty acid. So we might ask you to identify this right here, and on your multiple choice, you might have a fatty acid tail. It is a fatty acid tail. Okay, but what happens if fatty acid tail isn't on one of the choices? But a saturated fatty acid is. Well, then you would pick that. All right? So, in general, that is a fatty acid, but what kind? It is a saturated fatty acid. Each carbon has 4 bonds around it, all the way to the very end of the chain. Okay? Well, if you have saturated fatty acids, you probably have to have what? That's right, unsaturated fatty acids. I'm just going to abbreviate that, FA, fatty acid. Well, where does that come in? Well, sometimes a couple hydrogens are missing. Okay? All right. So we're missing those 2 hydrogens. That means this carbon and this carbon do not have 4 bonds. This carbon has 1, 2, 3. This carbon has 1, 2, 3. But in chemistry, we can fix that and maintain our structure. We simply put a double carbon bond between this carbon and this carbon. So when I say, does every carbon have 4 bonds around it? This one has 1, 2, 3, 4. This one has 1, 2, 3, 4. This one has 1, 2, 3, 4. Carbon has a unique ability to form single carbon bonds, double carbon bonds, even triple carbon bonds. But again, which one is the more stable? Single. Double is less stable. All right? So what gives this away right here? If I were to ask you, what is this? You go, oh, that's an unsaturated fatty acid, and you would be correct. But if they don't have unsaturated fatty acid, and they have fatty acid or fatty acid tail, you can pick those. All right? Now, if I did construct my 3 tails out. This whole entire thing would be a glycerol head with 3 fatty acid tails that are either saturated or unsaturated, and we call that, since there's 3 of them, we call this a triglyceride. Looking in my notes here. Okay. Got this thing blinking, so I'm just trying to use the camera so it doesn't turn off on us. All right. Use the telephoto there a couple times. Let's erase this. Oops. Still on lipids, otherwise known as fats. Okay. Let me grab my marbles. Okay, remember your glycerol head? Bam, right there. 1, 2, 3. Okay, OH, 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 dehydration synthesis to put the tails on. So this right here is your glycerol, okay? Glycerol. Okay, that's the glycerol head. Then guess what this is? That is a fatty acid. Okay, fatty acid. Okay, long chain of hydrocarbons. Single bond, single bond, single bond, single bond, single, 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 single. What kind of fatty acid would this be? A saturated fatty acid. Okay? A saturated fatty acid. Single, single, single. Ooh, look what happened right here. A double bond. Missing a hydrogen there. Okay? So a double bond simply means this is not a saturated fatty acid, all right? This is an unsaturated fatty acid. Okay. And if you have your 3 carbon heads right here, 1, 2, 3, and then you have 1, 2, 3 fatty acid tails, your triglyceride. Okay, this is your triglyceride. Okay? I'm going to go throw these back in the box. Okay, a note I want to make, maybe I should have made it sooner, but your textbook, and there's several versions of the textbook that you can get. when you're looking at carbohydrates and whatnot, Chapter 2 is chemistry, and one of the things I wanted to show you right here is that on page 39, Chapter 2, figure 215, also figure 214, okay, so I'm pretty sure those figures close to each other in all the books, 215 and 214, shows the two-dimensional pictures of the molecules that we've just been doing. Turn the page, and this was our 3 carbon head and our 3 fatty acid tails, and this is our triglyceride or neutral fat. Okay, we're going to go over the cholesterol molecule, which is right here, or a steroid. Okay, steroid molecule. And you can find those in your book. All right? All right. Here is your cholesterol molecule, It has 4 interconnecting rings that are 6, 6, 6, and then the last one is 5. So you have 6 rings right here. And then this little tail, little hydrocarbon tail, 1 oxygen. This is your cholesterol molecule. What does the cholesterol molecule do? It does a lot of things. One of the things that cholesterol makes is bile. Bile is stored in your gallbladder. Don't make that mistake. Bile is made in the liver, in the hepatocytes, which are liver cells. It then trickles down and is stored in a thumb-size sac called your gallbladder. Your lecture people next year might want to try to catch you and say which of the following is where bile is made, and they'll put A, gallbladder. 50% of you are going to fall for that. Make sure you don't do that. It's just stored in and released from the gallbladder. But sterol molecule can also make steroid hormones. Okay. Cholesterol is inserted into the cell membrane to give it structure, because the cell membrane is made up of phospholipids, which we'll be talking about. This right here, just the rings that I was telling you. There's the first ring, second ring, third ring, 6 carbon rings, and a 5. This is what they call the steroid skeleton. This steroid skeleton can make steroid hormones, testosterone for men, and estrogen for women from this molecule right here. Let me mess with my camera. In and out. See that it doesn't turn off. That way it thinks it's being used. Maybe you actually have to move it. I don't know. Yeah, I'll get it right there. All right. We're ready to end up with peptides. Let me bring my peptides. Here we go. All right. Gonna have to use all my colors for this right here. Okay. Roman numeral 3. Proteins. Proteins are made up of, I got ahead of myself, amino acids, of which there are 20 different amino acids in the body. Okay, 20 different amino acids in the body. From those 20 amino acids, you can make millions upon millions of different proteins. All you have to do is have different sequences of those 20 different amino acids, and that will give you all the different functional proteins that operate in your body. Just like we have 24 letters in the alphabet, And with just 24 letters, we can make millions of words in English, Spanish, French, German, all kinds of different languages. What's that? You said that there's 26 letters in the alphabet? No, there's only 24 because E.T. went home. E.T. went, never mind. All right, amino acids are also known as peptides. You can have a monopeptide, you can have a dipeptide, you can even have a tripeptide, and you can even have what's called a polypeptide. Okay, mono meaning one single peptide, which we're going to draw here in just a second. Okay, once you can make a monopeptide, you can put two monos and make a di. Okay, you can add one more mono to a di and you can make a tri. And polypeptides are when you have many peptides strung together. Since this is talking about proteins, how many peptides does it take to have a protein? You need to have more than 50. So, as soon as you hit 51 peptides, which would definitely be a polypeptide, 51 is originally a protein. Okay? All right. Anything with less than 51, we just call them polypeptides. All right? So let's draw our peptide. NH2. Okay? Our amine group, one nitrogen, two hydrogens. Okay? So that would look something like this. Okay? A single nitrogen with two hydrogens. Okay? With a central carbon. Now, as soon as you get that carbon, you have to draw 4 bonds around it. Okay? And we get our acid group, COOH. Our amine group, our acid group. Okay? At the bottom here, we have what's called our obligatory hydrogen. Okay? It's always going to be a hydrogen. So every single peptide has these 4 things in common. An amine group, a carbon, central carbon, an acid group, the obligatory hydrogen, and something called the R group. Okay? This is our peptide. Okay, let me make sure that's really focused. I'll zoom right in on that guy. Right there. Okay? That is what we wanted. Okay? Let me pull back because I'm going to hold up a there we go. All right. So a single peptide looks like this. Here is our NH2, our amine group, a central carbon, and down on the bottom we have a hydrogen, our COOH, our acid group right here. So amine group, acid group, central carbon, obligatory hydrogen, and R. What the heck is R? Okay. Question is, the R is what gives an amino acid its name. How many different R groups do you think there are? You'd be right if you said 20. There are 20 different things that we can put here. Okay? In algebra, you might just call that X. Okay? For our purposes, there are only 2 specific amino acids you have to know by name. If in that R position we put an H, okay, this becomes glycine. Notice the end of our name is ine. Ene signifies the fact that you have a peptide, an amino acid, ine. 18 of the 20 amino acids in our body all end in ine. Glycine, threonine, cyranine, alanine, everything ends in ene. There are 2 amino acids that have the word acid, glutamic acid and trying to think of the other one, glutamic acid and some other acid. We don't need to know that. These prefixes and suffixes are good to know. All of our sugars, did you notice what all sugars end in? All sugars end in OSE, glucose, fructose, maltose, OSE. Os means you have a carb. Ene means you're talking about a protein more than likely, or an amino acid. Okay? Fats don't have that designation. Okay, it'd be cool if it did, but not really. All right. So this is glycine. What happens if I put a methyl group there? Okay? Another reason to talk about methyl. Okay? A single hydrogen gives us glycine. CH3 gives us alanine. Okay? And let me show you a glycine. The original one that I showed you, here is the central carbon, okay, amine acid, central carbon right here on the bottom, hydrogen in the R position, that's a hydrogen. This is a peptide or a glycine or an amino acid, okay, need to know all those, okay. CH3, here is my amine group, here is my carboxyl or acid group, my central carbon, the obligatory hydrogen, and in the R position, CH3, alanine. Okay? If we were to ask you, identify this, what do you think this is? Okay, let's go from left to right. An amine group, central carbon, acid group, another amine group, another central carbon, another acid group, another obligatory hydrogen, and a hydrogen in the R group. So we have 2 hydrogens in the R group. That means we have glycine and glycine. We don't have a name for this, but we would call this not a monopeptide or a peptide. This is called a dipeptide. Pretty easy when you see 2 blue marbles. Dipeptide. What happens if you see 3 marbles? Tripeptide. Exactly right. All right. So I'm going to do these not in color.